Lesson 2. The blood and the blood clotting process. Objectives of the lesson. Focus points. Identify and describe the different components of the blood. State the function of the different components and explain the blood clotting process.
Do you know what a vampire is after when it bites? It only wants some of your cells, called erythrocytes. Well, a red blood cell is a tiny disc with a dent at the center and hemoglobin near the surface, so oxygen can easily leave or enter. The function of these red blood cells, or corpuscles, is to carry the oxygen to your tissues and muscles. Well, a red blood cell is a tiny disc with a dent at the center and hemoglobin near the surface, so oxygen can easily leave or enter. Most of blood is plasma, and if you want to know what that is, it's just stuff dissolved in H2O. There are other cells in blood known as leukocytes. They help fight infection and are big and white. Well, a red blood cell is a tiny disc with a dent at the center and hemoglobin near the surface, so oxygen can easily leave or enter. If you're cut, you can get all the plasma you need, but without some platelets, you're just going to bleed. Well, a red blood cell is a tiny disc with a dent at the center and hemoglobin near the surface, so oxygen can easily leave or enter. La 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 la. La 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 la. La 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 la. La 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 la.
Don't take this the wrong way, but you're pretty replaceable. When it comes to your body, science has figured out how to hack, synthesize, or replace a surprising amount of its parts and processes. We have implants to keep heartbeats steady and steel rods to mimic bones. We've got drugs that can replace hormones and antibiotics to cover for your immune system. And pretty soon you'll be able to just 3D print a new ear if you need one, really. But one thing we absolutely cannot manufacture, despite what True Blood would have you believe, is blood. And yet blood is a thing that we all need. And sometimes, because of injury or illness, we need extra blood. In fact, every 2 seconds, someone in the US needs a blood transfusion. This could be a victim of a car accident, someone undergoing surgery, or a cancer patient who needs new blood to maintain their health during chemotherapy. And because we can't grow it on trees or make it in a lab or even store it for all that long, the blood that people need, nearly 16 million pints a year in the US, has to come from people who have donated it. So, let's talk blood, shall we? The meal of choice for vampires and female mosquitoes, blood is red, sticky, salty, and kind of metallic tasting. tasting. It is indeed thicker than water and super viscous, which is why Hitchcock used chocolate syrup as a stand-in a certain classic shower scene. For most purposes, blood comes in 8 different types, and it accounts for about 8% of your body weight. You might remember from our episodes on tissues that blood is a type of connective tissue, which means it's made of living cells suspended in a non-living matrix, which in this case is the fluid ground substance called plasma. And of course, one of blood's main missions is to transport and distribute oxygen and nutrients and waste products and hormones around the body. But it also helps maintain and regulate body temperature, pH levels, and the volume of fluids in your body. Plus, it protects you from infection and from the loss of blood itself. Perhaps second only to your brain, your blood is the one component of your body that we haven't figured out how to reproduce, synthesize, or imitate. It's a part of you that is literally irreplaceable. It's Saturday and you feel like doing a good deed, so you head over to your local Red Cross for a blood drive. You get your finger pricked and then somebody directs you toward a lounge chair, swabs your inner elbow with alcohol, and then comes at you with a hollow needle. Once the bag is full, they usually take about a pint, you get unhooked and grab a cookie and a juice to replace the blood sugar you lost, and the whole process takes around 20 minutes. But for your blood, the day is just beginning. Soon it will be taken to a lab where it'll be tested for infectious diseases and separated into different parts before heading out to hospitals. So hold up. What exactly do I mean by different parts? Well, the blood that flows from your arm into that bag is whole blood, a mixture of cells and cell fragments called formed elements along with water and lots of dissolved molecules. A patient who needs a transfusion may only need some of those things and not others, so the parts are separated. Once your blood makes it to a lab, technicians put it in a centrifuge which spins it around fast enough to send the heavier components to the bottom of the tubes and bring the less dense elements to the top. In the centrifuge, 3 distinct layers emerge. Down at the bottom, you got a heavy red layer of erythrocytes, or red blood cells that carry oxygen and carbon dioxide. They make up about 45% of your total blood volume. Then you got this thin little whitish layer in the middle. Those are your warriors, the leukocytes, or white blood cells that defend your body from toxins and foreign microbes. And there are also the cell fragments called platelets, which help with blood clotting and make up less than 1% of your blood. Finally, up at the top, you see the yellowish plasma, which accounts for about 55% of your blood volume. Plasma is actually 90% water, but the other 10% is chock full of 100 different solutes, including proteins, electrolytes, gases, hormones, and waste products. The most abundant of these solutes are electrolytes, which you may have heard of as the secret ingredient in sports drinks, but they're really just positively charged cations like calcium, sodium, and potassium, and negatively charged anions like phosphate, sulfate, and bicarbonate. Together, these ions help regulate your blood's chemistry, maintaining its pH levels and proper osmotic pressure, and allowing other tissues to do their jobs, like making muscles contract and sending action potentials. But when measured by weight, the bulk of the solutes in your blood are really the plasma proteins. Most of these proteins, like albumin and alpha and beta globulins, are made by the liver and do things like balance the osmotic pressure between the blood and surrounding tissues and transport lipids and ions. Others run defense for you, like the gamma globulin antibodies that are released by plasma cells during an immune response, or fibrinogen proteins, which are vital to forming blood clots and stopping bleeding. Alright, bleeding. I want to talk about that. Because for the very reason that I mentioned at the beginning, that we can't replace your blood with some synthetic wonder fluid, the last thing that your circulatory system wants is for you to fritter away your blood in some sidewalk scrape or kitchen accident. So it has a whole system in place to prevent you from losing too much of it through a process known as hemostasis. So imagine you're slicing up a nice garlic cheese bagel one morning and you lacerate the distal phalanx of your pollex. In other words, you cut the tip of your thumb. And now you're bleeding all over your breakfast. At the very first sign of rupture, the blood vessel actually constricts itself to slow the flow of blood through it. Then little cell fragments called platelets gather at the site of the injury, creating a plug that dams the breach and keeps the blood from leaking further. Now, these free-floating platelets don't clump together during regular circulation, that would be terrible, but when the endothelial cells lining a blood vessel wall tear, the underlying collagen fibers are suddenly exposed, and they chemically react with the platelets, turning them all sticky and glue-like at the scene of the injury. But that platelet blood plug still isn't as strong as it could be. It needs reinforcement to complete the clotting process. This reinforcement comes in the form of fibrin threads, protein strands that join together to make a sort of mesh that traps the platelets and blood cells. Eventually, the threads actually pull the opposite sides of the wound together to close the vessel wall so the endothelial cells can be replaced. Over a few days, the blood vessel heals and the blood clot dissolves, or at least that's how it's supposed to happen. People who suffer from disorders related to hemostasis may have trouble with unwanted clotting or the inability to form clots. In the family of disorders known as hemophilia, a patient can usually complete the first 2 steps of hemostasis just fine, but they can't make an effective fibrin clot. So it's not that they bleed more than anyone else, it's just that they bleed longer, which I guess means that they bleed more. As a result, they may need frequent blood transfusions throughout their lifetime. Which brings me right back around to that Saturday morning blood drive. Another thing you're gonna need to know before you give blood is what type you have. Do you have A, B, AB, or O? These different types all do the job equally well, they just sort of have a different flavor related to your immune system. All the cells in your body have a plasma membrane with specialized glycoprotein markers on them that act like name tags or labels, sort of like, this cell is property of Hank. These markers are your antigens, and your body's immune system is totally fine with your particular antigens, but if it detects antigens from someone else's cells, including viruses or bacteria, then it'll send out antibodies to bind to those markers, often to tag them for destruction by the immune system. Your red blood cells have specialized antigens on them called agglutinogens that activate antibodies that work by binding invading cells to each other, which causes coagulation, or the clumping of blood. Which agglutinogens you have on your erythrocytes defines your blood type, but they're classified in 2 different ways. In the most important blood classification, the kind people are most familiar with, there are only 2 kinds of agglutinogens, simply A and B, and your blood can either have one or both or neither of these molecules. So the name of your blood type refers to what kind of agglutinogens you have or don't have. A type has A antigens, B type has B, AB has both, and O has neither. So why do you need to know what type you are before you give or receive blood? Well, like I mentioned, if you have either of these antigens, your body will be fine with it because it doesn't produce any antibodies that label it for attack. So if you don't have a particular antigen on your blood cells, say the type B, then you do have antibodies that are going to label those B antigens for attack should they enter your space. So AB type folks are called universal recipients because they have both antigens and therefore no antibodies for either. So they can accept A or B or AB or O blood. Meanwhile, O type doesn't have A or B antigens, so those folks have antibodies for both. That means that they can only accept other O blood. And yet that lack of antigens means that type O blood can mix with other types of blood without getting attacked, which is why it's known as the universal donor. But just to complicate things a little bit more, you've got a whole other set of antigens with totally different protocol. These are your rhesus or Rh antigens, named after the species of monkey they were first identified in. Much like A and B, you either have the Rh antigens, in which case you're Rh positive, or you don't and are Rh negative. Most of the population is Rh positive, so they don't have the anti-Rh antibodies, which means they can accept either positive or negative blood, but negative types should stick to just the Rh negative blood. And since the presence of AB antigens is controlled by different genes than the Rh ones, we end up with 8 different blood types, 4 separate groups, each with 2 variations. And now, hopefully, you understand why it's so hard to replace blood, and why true blood is not true. I've not actually ever seen that show. Along the way, you also learn the basic components of blood, including erythrocytes, leukocytes, platelets, and plasma, as well as the basic process of hemostasis that stops bleeding, and how antigens are responsible for the blood type that you have. Thanks to all of our Patreon patrons who make Crash Course possible through their monthly contributions. If you like Crash Course and want to help us keep making it for free for everyone in the world, you can go to patreon.com/crashcourse. Also, a big thank you to Brian Drexler for co-sponsoring this episode. Crash Course Anatomy and Physiology is filmed in the Dr. Cheryl C. Kinney Crash Course studio. This episode was written by Kathleen Yale. The script was edited by Blake DiPestino, and our consultant is Dr. Brandon Jackson. It was directed and edited by Nicole Sweeney. The sound design was by Michael Aranda, and our graphics team is Thought Cafe.
First, what is blood? Blood is a liquid tissue and circulating medium of cardiovascular system. Blood is 8% of the human body weight. Adult has 5 liters of blood. The main function of blood is to transport materials and fight infections. There are 2 components of blood, the plasma and the corpuscles, or the formed elements.
Wave Beats Music.
Plasma. It is a liquid portion of the blood. 55% of the blood weight is plasma. It is a straw-colored fluid consisting of 92% of water and 8% of blood proteins and a trace amount of organic materials. Corpuscles. The corpuscles or formed elements suspended in the plasma comprise 45% of the blood by weight. There are three formed elements: erythrocytes or the red blood cells, leukocytes or white blood cells, and the thrombocytes or the blood platelets. These formed elements are synthesized in the red bone marrow of flat and long bones.
Wave Beats Music.
Erythrocytes. Erythrocytes are the red blood cells. The red color is attributed to the pigment hemoglobin, an iron-containing molecule that can bind with oxygen. This is the reason why red blood cells can transport gases through the body. Red blood cells are biconcave in shape, elastic, and unnucleated. Leukocytes, or white blood cells, are described as the soldiers of the body because their function is to defend the body against both infectious disease and foreign materials by secreting antibodies. Unlike red blood cells, white blood cells have indefinite shapes, and they are nucleated. The shapes of their nuclei and the presence or absence of granules in their cytoplasm are used in identifying white blood cells. There are two divisions of leukocytes: granulocytes and agranulocytes. Granulocytes are the white blood cells with granules, and agranulocytes are the white blood cells without granules. Thrombocytes. The thrombocytes are the blood platelets. Compared to red blood cells, they are not true cells. They are only remnants of the mother cell in the myeloid tissue. Blood platelets are irregular in shape, and their important attribute is they disintegrate upon contact with air. This is the reason why even in fresh blood samples, platelets cannot be observed. Such attribute is significant since upon disintegration, the blood clotting process begins.
Do you remember tripping over as a kid and cutting your knee, and then magically the bleeding suddenly stops? Or have you ever wondered why this happens? Our body is very clever and produces substances to seal off the open wound. In this video, we are going to look at how and why blood clots. Clotting prevents us from bleeding to death, and also protects us from nasty disease-causing organisms called pathogens entering our body. When we injure ourselves, we can damage the blood vessels just below our skin. This can cause blood to either leak into our tissue and form a bruise, or if the skin is broken, to openly bleed. To prevent these horrible things happening, blood clotting occurs. So let's have a look at how this happens. Starting with platelets. These are tiny cell fragments that float around in our blood. When a blood vessel is damaged, collagen is exposed. This collagen exposure attracts platelets to the injured area. The platelets then stick together to form a plug. They've quickly built a barrier that stops us from losing too much blood. However, this platelet plug isn't that strong, so it needs to be made stronger. A protein called fibrin does exactly that. In our blood, there are soluble fibrinogen proteins. At a wound, these soluble fibrinogen proteins are exposed to outside chemicals that aren't normally found in blood vessels. Like with the collagen and platelets, this exposure causes the fibrinogen proteins to turn into sticky fibrin fibers. The fibrin fibers form a mesh, holding all the platelets together and making a much stronger clot. More platelets, red blood cells, and other components in our blood all get stuck to the mesh, making it even stronger. This clot stops more blood escaping the body, and also prevents nasty pathogens entering our body from the outside. The clot develops into a scab, which protects the wound as it heals, and new layers of skin form underneath. So really, all a scab is just platelets trapped in the fibrin mesh. It is like putting a plug in a sink. When the new skin is fully formed, the scab will fall off, revealing the lovely brand new skin underneath. A final thing to note, remember that I said the exposure to the collagen causes the platelets to stick together, and the exposure to the outside chemicals causes the fibrinogen to turn into sticky fibrin. Both the platelets and the fibrin only become sticky after the exposure. This means in a normal, healthy blood vessel, they won't randomly cause clots. So from this video, you should understand the magic of blood clotting. Platelets are first to the scene, closely followed by the fibrin fibers. Our blood clots so that we don't lose a lot of blood, and don't let lots of nasty pathogens into our body if we cut ourselves.
If you liked the video, give it a thumbs up, and don't forget to subscribe. Comment below if you have any questions. Why not check out our FuseSchool app as well? Until next time.
The blood clotting process. The blood clotting process is an adaptation that stops the bleeding process due to an injury. Depending on the severity of the injury, the blood should clot within 2 minutes when a cut or an injury occurs. In the absence of the blood clotting process, bleeding continues and it can cause the loss of life. What are the steps in blood clotting process? First, when there is a cut, blood oozes out, releasing the plasma and blood corpuscles. The thrombocytes disintegrate upon contact with air. Next, the disintegration of the platelets causes the change of the inactive blood proteins, which is the thromboplastinogen to thromboplastin, an active chemical substance. Next, the thromboplastin plus calcium ions will catalyze the conversion of another inactive chemical substance, which is the prothrombin to thrombin. Next, thrombin catalyzes the change of fibrinogen, a soluble blood protein, into its insoluble form called fibrin threads. Next, fibrin threads form a network over the injured part and seal it to prevent more blood cells from coming out until a red mass forms on top of it. And lastly, after some time, the clot shrinks and straw-colored fluid called serum exudes out of the injured part. This serum is a blood plasma which lacks blood protein fibrinogen in it. And for the summary, the blood is the medium of circulation. It is composed of plasma or a liquid part and the formed elements. The formed elements are the erythrocytes, leukocytes, and thrombocytes. Erythrocytes are the red blood cells, leukocytes are the white blood cells, and lastly, the thrombocytes are the blood platelets. The blood clotting process consists of series of events involving the erythrocytes.
Wave Beats Music.